Would you please give it up for a Wednesday afternoon for the great Dr. Kate Michie.
Thanks, team. I'm assuming you guys are comfortable for another lecture. If you fall asleep, that's totally fine. I can understand it. I'm hoping this lecture now will tell you why this is so exciting. So the first one was really, what's the background, what's the field, how does the technology sort of work, so that you don't need to be afraid of it and you understand the context. But now this is, I think, the really exciting stuff, because this is the future and this is the new stuff. Okay, so this is from 2022, so it's old data now. But this is the first papers describing AlphaFold, and by the end of 2022, or beginning of 2022 moving up, you can see how many articles were citing AlphaFold. So this was actually adopted by the scientific community really, really quickly. So what do we know about proteins before AlphaFold? So if you have a look at humans in this chart here, the blue is actually how many structures we knew are in that organism beforehand, experimentally solved, so things that we had good quality data for. And then there's some other methods we have that give you some information, but it's not very good, but it's still some. And then there's knowledge from AlphaFold. And you can see, so the 100%'s up here, and this is humans. So you can see that already in 2022, that AlphaFold was contributing at least half of the information that we knew about proteins in humans. That's 1 year. AlphaFold came out July 2021. So this is why this is game-changing for people in sort of biology. And you can look at, these are all kind of other model organisms, so there's yeast and Staph aureus, so this is, bacterial infections, tuberculosis. So these are common pathogens. Obviously, knowing everything we can about those are really important. Like TB's got increasing antibacterial resistance. It's a major threat to human health. And look how many structures we understood, or what we knew about the proteins from TB. Not very much. So solving protein structures has been a huge hurdle in biology. So that's why this is such a game changer. So one thing I didn't tell you about is that AlphaFold has some built-in metrics. It can evaluate how well it thinks it's done. So it outputs two metrics. One's called PLDDT and one's called PAE. We don't need to go into the details about it. There's lots of good help documents, and if someone's really interested in having a go of AlphaFold, there are actually free resources, and I can point you into directions to easy documentation to help you understand what those metrics mean. But those metrics enable specialists and non-specialists to evaluate the output of AlphaFold, so we can see whether it's done a good job. Sometimes the metrics we're learning aren't perfect, but most of the time they're pretty good. So the main focus for me as a structural biologist is to help people learn to understand how to read those metrics so they can actually evaluate how AlphaFold has evaluated their system and how they can use the models that are coming out. So now I want to show you real-world cases of AlphaFold, most of them which I've worked personally with people at UNSW or around Australia, and a couple of international ones, just to show you the variety of ways that this can be used. There's lots more, right? There's thousands of reports of this now, so I want you to kind of just be aware of the different ways that may not be completely obvious. Okay, so this was our very, very, very first experimental use of AlphaFold. So this, what you see on the screen, is a protein structure from HTLV. So it's the oldest known retrovirus. It's related to HIV, and it's the most carcinogenic virus known. And it's endemic in the indigenous population in Australia and in Southeast Asia. So it causes cancer, and we have no treatment for it. It's like HIV, right? It's a terrible, terrible virus. A retrovirus actually inserts its genomic code into your genome, so it makes them really hard to treat. So the researcher who works on this at UNSW is an HIV expert, and he was particularly focused on HTLV because it's endemic in Australia and it's an important disease. This particular protein that you're looking at is the capsid protein. So this is the shell, the skin that goes around the virus, and no one had been able to solve that experimentally before. So we had very little to lose at this stage. People have been trying to do it for sort of 30 years and failed, and the literature was, you can't solve the structure of the HTLV capsid protein, it don't work. So my colleague came to me with that when AlphaFold first came out and said, can we predict the structure of the HTLV capsid protein? I went, sure, no worries. Chucked it into AlphaFold, out came the answer, and I gave it to him. He goes, oh, that's interesting. It looks like it's made of 2 domains. I'll just cut the protein here when we make it experimentally and see if that works. And immediately, he got crystals. So for crystallography, that's the rate limiting step. Can you make the protein? Can you crystallize it? If you don't get crystals, you haven't even hit first base. So he got crystals, they diffracted to very high resolution, which just tells us we've got very, very good crystals. And he was able to solve the structure of both of the components of the protein. And the difference here, and I apologise to anyone who's red-green colourblind because my colleague prepared this slide and he is red-green colourblind. He's not red-green colourblind. He said he doesn't understand that we shouldn't use red and green together. So I apologise to anyone who is. For anyone who is red-green colourblind, the arrows indicate the difference between the calculated prediction and the experimental version. So this was in the first few months of AlphaFold coming out, that we had already experimentally validated using a structure that has never ever been seen before, so AlphaFold couldn't train on it, and we solved the structure of the protein.
So can I just clarify? So those 2 arrows are the
only differences. Yeah. So this showed to me and to him, who were both structural biologists, that this is a holy cats moment. Like, something's happening here, right? This is scary. It's a whole discipline of people who've spent 10 years trying to solve one crystal structure, and all of a sudden you can calculate it overnight. So that was a bit of a shock. So why is this useful? Okay, so he's an expert in the HIV field. This particular structure is really important because it shows us that this particular retrovirus has the same pocket that the best new HIV virus, Lenacapavir, has. This particular drug is really important because it can be used very early as a prophylactic, so before anyone's infected, and it lasts for a very long time, so you only need to treat people twice. And it suggests that the HTLV virus has the same druggable pocket. So it means that this avenue of approach is probably amenable to this particular virus. So for the HTLV field, who are trying to stop cancer, this is a really big step forward. And that all happened from one AlphaFold structure, that we were able to crystallize it, something that we couldn't crystallize before, based on understanding how the protein looked. So the other thing that I'm not telling you about in the slides is, if you remember that really ugly looking equation I told you about, how crystallography has these 2 components. You need the diffraction pattern, and you also need this weird thing called the phases to get the calculated structure. So we got really high level diffraction from this, but we didn't have the phases. Now the phases are telling us we can steal them from molecules that look the same. And so my colleague tried to steal those phases from other virus structures that we thought would be similar, and it didn't work. So we took the phases that were calculated from the AlphaFold structure and used those to solve the experimental data. So all of a sudden, a whole bunch of experimental problems that we haven't been able to solve because we couldn't get the phases were actually able to be solved experimentally using the phases from AlphaFold structures. This step alone changed crystallography. So getting phases was really hard. And so all of a sudden, that's now possible for a whole number of cases that we could crystallize but we could never solve. So it's a mathematical problem, but this was really groundbreaking for us. Like, we did not expect that. And it actually took us a long time to realise that. The first thing my colleague tried to do was soak in a whole bunch of heavy metal ions, like mercury, into his crystals to try to get the phases, because that's the old-fashioned method, was to try to do that. And that totally failed. And he's pulling his hair out. I said, well, why don't you try the phases from AlphaFold? That afternoon he had the structure. Like, it's a big game changer. So now we've got a drug handle on this particular virus, and the field now is moving ahead. So here's the personalised medicine question that I think Chris was talking about before. So the lady in the office next door is a clinical geneticist. She works with children with muscular dystrophies. These are really horrible muscular diseases. They're often bespoke mutations, so the patient has a mutation that's very, very rarely seen. It doesn't look similar to what anyone else has. The typical approach is you sequence the mum and the dad, and you sequence the kid, and you try to find the difference. And she comes to me often with proteins that have a single change to ask me, is this likely to be causing a problem? And in this particular case, there's a little pink blob in there somewhere. If you see it when it turns around, there's a pink blob right up here. It's a single amino acid change in this particular case. And it looks like that's the single change that's causing the mutation in this particular patient. So these are the patients that haven't often had diagnoses for decades. So some of them are young children, some of them grow up to adults, they're still alive but they're very sick, and they haven't been able to diagnose what's wrong with them. So using this technique from AlphaFold data, from structures that we've never been able to see before, we can now predict whether that particular mutation is the cause. And if we can identify which protein it is, we can often understand which pathway inside the cell it does, so that we can find the right sort of therapeutic, and we know how to treat them. So we now already have clinic, like therapeutic applications, from 6 months of identifying the change in AlphaFold. So this is actually flowing into the lab, into the clinic now in patients in Sydney. This is a game changer. And this was unexpected. So this is for fundamental science. So I work with a lot of people that work on these weird organisms that live in stromatolites in Shark Bay in Western Australia. So these weird old geological things that grow in high salt. And I have colleagues that work on things that grow out of deep sea hydrothermal vents. And if anyone's watched those David Attenborough movies of deep sea black smokers with weird creatures living at the bottom of the abyss, I love that kind of stuff. These proteins that you see here are actually folded and they've never been seen before. They have a completely unique fold. It's never been seen in any organism previously. And we're able to do this now from sequencing data. We can now fold the entire genome of these organisms in a couple of days. So I haven't told you here, the reason I can do it in a couple of days is because we have some other computer codes that developed after AlphaFold, which can fold things faster than AlphaFold. Not quite as accurately, but we can fold them really quickly. And when we find something really exciting, we can then refold it much more carefully with AlphaFold. So this has been really helpful because now we can start searching through a whole proteome. It's like instead of the genome, I'm sure you've used to the phrase genome is your DNA, the proteome is all of your proteins, or all of an organism's proteins. So this helps us identify a whole bunch of novel pathways. So lots of microbes have really cool, weird metabolic processes. They've got enzymes or proteins that can do chemistry on really weird chemicals because they live in weird environments. And so those things are super useful for biotech and industry. If you look at your washing detergent, you might find it says enzymes on it. So an enzyme that they're using in that case is a protein that breaks down another chemistry, and those things are used by biotech to increase or improve performance of compounds. So we use those a lot, and finding them from really odd organisms is a huge advantage in biotech. So we can fold a whole organism now. And then this was something that we didn't expect. So researchers, once AlphaFold was made public, very quickly tried gluing proteins together with long chains of amino acids. So we get protein A and protein B, and they've just put in a long chain of amino acids and told AlphaFold to fold that. What would happen? Would it calculate two separate structures? What it did is it calculated two separate structures, but in the cases where those two proteins interacted in vivo, so inside the cell, it actually folded them together as though they were interacting. So for the first time, we can predict in silico a protein-protein interaction. Now that method in the lab is really hard. So all of a sudden now, through big compute, we can start to identify which proteins bind to each other. This was unexpected. And that was actually found by researchers who Google had released the code to. We'd all were playing with it. So very quickly, Google then started to retrain AlphaFold specifically on multimeric structures to get better at this task, and then released a new version of AlphaFold that was specifically trained to handle multimers. So multimers just mean more than one. It could be one of the same protein or two different proteins. And that can actually extend to two different proteins from different organisms. So a host-pathogen interaction. So me and tuberculosis. That interaction's harder for us to do because we have to mine the evolutionary data between two different organisms, but it's possible. That space is improving very quickly, so there's lots of jiggery-pokery we have to do at the back end with the multiple sequence alignments to make that work. But it's giving us an opportunity to start probing protein-protein interactions to understand how the cell assembles all of its components. And that was totally unexpected. So this is a particular case from some medics at UNSW. They work with macromolecular degeneration. So they have a special apparatus that they can electroporate someone's eyeball. Sounds gruesome. But they can use that to uptake gene technology. And they know with macromolecular degeneration that there's a particular receptor, which I'm showing here on the screen, which we didn't have the structure for in the dimer form. And they wanted to know how they can stop this receptor turning on. And the receptor turns on when it binds together as two forms. So you've got one protein here and the other protein there. So it's the two of them together. And the AlphaFold structure shows us the region of the protein where they interact. So we were able to design a gene therapy construct, which is just this little blue bit. And if you can overexpress that in the cell, it binds to the full-length protein and blocks it from dimerizing. It's that simple. It's like a drug. You just make a small protein that binds to that location, it stops the two things coming together, and you stop macromolecular degeneration. So we were able to do that with AlphaFold. They've put it through animal models at the beginning of the year, and all 8 rabbits with macromolecular degeneration have been cured. That's wild. I didn't expect that. I don't think they expected that. So here's one that's happening across the field. So this is a viral case. This was the way the field thought a protein actually folded. So it's often common in structural biology, proteins are so big and they're difficult to work with, that we often cut them into smaller pieces to work with them. And this particular piece had been cut down and they thought this is what the protein structure looks like. But AlphaFold shows us that when you fold the whole protein in context, not cut, that the structure that it forms is very different. So this just threw 20 years of a whole research field out. Overnight, changed the game. Now they know this is what they need to work with. So one calculation that probably cost about $400 of cloud time threw away 20 years of full-time research from many, many research groups. That's a bit of a wow moment, isn't it? Here's an example. I worked for head of medicine. He came to me, he said, he works on malaria. He goes, I've got this weird little protein. AlphaFold says it's not very interesting. Yeah, I agree. It doesn't look very interesting, does it? AlphaFold says it doesn't really interact with much. But because I'm a structural biologist, I go, oh, the sequence of that protein, it's those hydrophobic amino acids I was talking about before, ages back. Remember that? It looks a bit sticky to me. It looks like it belongs in a membrane. Membrane proteins like to be bigger. It's probably not just a membrane protein. Let's see what happens if we do a multimer. AlphaFold can very confidently predict that this protein forms a channel in the membrane. So this is a rotation. You can look down, there's a hole in the middle, so it forms a pore. It shows a charged channel. So this is quite common that we know in ion channel structures, that you have a little hydrophobic gate. So the white indicates that it's hydrophobic, and the red and the blue means positively and negatively charged. So I've drawn in a little membrane here of how we think it fits into the membrane, and that it has all the characteristics that we would expect of an ion channel. And this was great because this protein had been previously thrown away as a potential target for a new therapeutic because they assumed it was such a small protein, how could it possibly be the target of this drug? It must be a false positive. This drug only worked at a very specific phase in the malaria's parasite's life cycle, which makes complete sense now that it's an ion channel. And so that research group has completely redirected their entire research to understand how the channel functions and to improve the drug binding of the channel in this particular way. So that was an unexpected $400 calculation that you would never ever have found easily before. Okay, so here's a cryo-EM structure. So this is using that new cryo-EM method. This was one I show because I spent my PhD working on these proteins back in the day. It's taken 30 years of research to build this complex up. This protein complex is important for building the cell division apparatus of bacteria. And why would you care about that? Well, bacteria can be pretty toxic. They can be pretty hurtful to you. There's also some good guys. But this particular one comes from Pseudomonas. Pseudomonas causes all sorts of really nasty infections. And so if you want to stop cells dividing, you need to pick on things that are involved with cell cycle, replication, division. If you can block division, you're going to kill the bacterium, right? So 30 years it took them to work out this structure. 8 hours, I can calculate a related organism structure that gives you a high level of confidence and high resolution so you can plan very directed experiments in the lab on a different organism. So as a research tool, it can totally accelerate the things you might do in the research lab when you're doing wet lab experiments. That's a big change, right? 30 years, 8 hours. I always felt really sad the moment I did this. Like I spent 4 years of my PhD and didn't get a lot out of it. 4 years and I did the whole thing, way more impressive than anything I achieved in my PhD, and I did the whole thing in 8 hours. I didn't even do it. I just put the code in and hit go and went and had a glass of wine, and out came this structure.
Sounds like a win to me, honestly.
Yeah. I mean, this is just, it's shocking. Most structural biologists are still coming to grips with this. So you can mine protein complexes, something I was saying before. So we've learned all sorts of things. All sorts of really unusual, unexpected interactions have been already discovered by mining protein-protein interactions using protein A and protein B in AlphaFold with a high confidence indicator from the metrics that it has. So it's not answering the question. You still need to do lots of validation, but it's enabling us to triage appropriate experiments really quickly. So it's making science cheaper and faster. And that was unexpected to me. And it's also being used by non-traditional, sort of not structural biologists. So the data is now really, really accessible and very, very useful for people that would never have looked at structural biology before. Because let's be honest, making protein crystals and solving structures was like brute force, and like you had to be stupid to do it, right? So it's making structures really easily available. All right, so I alluded at lunchtime to a few people that AlphaFold 3 was released. So in May last year, AlphaFold 3 was released. Now I keep AlphaFold 3 in a separate box because AlphaFold 3 has a different algorithm behind it. So while the name's the same, it works a little bit differently on the inside. So it's heaps faster than AlphaFold 2. So you can go to Google yourself, and I encourage you to do this in your own time. Go to Google AlphaFold 3 and you can access the web server and you can copy and paste a sequence of a protein and ask Google to calculate it, and it'll calculate it for you and you can see it straight away. And it takes a few minutes. So something that was taking us 48 hours can be done in a few minutes. And Google offers that for free. It can also now include nucleic acid. So DNA, RNA, it can include protein modifications. So these are things like sugars, other sorts of things that are glued onto the protein in the cell, and drugs. Wow. There's licensing issues. So big tech owns all of this code. So you can't use it if you're a company. Academics can use it, but if you're in a biotech company, you're not allowed to use it. So just because the code's been made open source so you can see it doesn't mean that you're allowed to use it. So this is causing all sorts of problems with IP in science because people are putting important things into it that they're effectively giving Google ownership of their IP by using it. So there's all sorts of other issues that you need to be mindful of. So there's now a whole bunch of other pieces of code that have been developed by other machine learning structural biologists that do the same task and are open source. Each one of those is often retrained to do a particular task. Some of them make some really cool changes that make them run faster, cheaper, use the GPU more efficiently, can use cheaper cards. So this space has really exploded. We run probably 10 different pieces of code at UNSW now. And that's, this piece of code was, released a year ago. And we're already running completely new pieces of code on top of it already. So here's some structures of nucleic acids. So these are RNA knots and repeat unit. Both of these are involved from researchers at UNSW. This is from the same researcher that works on the HTLV, showing that a particular RNA sequence forms a pseudoknot. That's basically just a structure that blocks it running into the ribosome and changes some of its control. So that's important for him to understand how the virus is making proteins. And the second one's involved in antibiotic resistance. So some of these structures are really important. We have a first case of a RNA abnormality that's causing genetic disease. So this is coming from the same team that we work on with muscular dystrophy. So this is in RNA this time, so it's not the protein. So there's a single change in the RNA sequence, and it totally changes the structure of the RNA. This particular mutation is unusual because there's these things called silent mutations. So there's a code change in the DNA that encodes for the same amino acid. So the protein result of that DNA would be the same, but there's a change in the DNA. And that particular change would be thrown away by any of the geneticists because it makes the same protein. But in this particular case, that change in the DNA gets copied into the RNA, and that changes the structure of the RNA, which changes how well that protein can be made. So for the first time, we have a case now that a genetic disease comes from a silent mutation. This will change genetics because mostly we would throw away those mutations as not likely to be a problem. But now we know that it can perturb nucleic acid structure, and that's a likely cause of disease now. So that's opening up a whole new ballpark of problems. Okay, so the problems, the programs aren't perfect, and I want to show you a few bloopers that AlphaFold makes. There are definitely cases where we have experimental structures and AlphaFold predicts a structure, and we have calculated the structures from lots of biophysical and structural methods, and we know that the structure is wrong. We do not know why in these cases sometimes it doesn't work. Still don't know. Sometimes it's a computer program, it will calculate an answer. So it's always going to be confident that it's calculated something. Just because it's calculated it doesn't mean it's right. So if we turn this protein around and we zoom in, you can see it's kind of evaded the laws of physics and it's put atoms on top of each other. That's a classic fail. It turns out this actually happens when it runs out of memory. So this happens in the GPU. So having a big GPU really, really matters. But we didn't understand that when we first started this. But if you didn't understand that this wasn't possible, or you didn't look at your structure carefully, you might use this structure and it's obviously got some big problems. So you need to be careful, right? So this is a tool that needs to be used carefully, but it can be very helpful. These are two proteins that we've calculated some multimers, so dimers, and we know these proteins cannot or do not interact. But AlphaFold has confidently built very strong and very high confidence indicators that these proteins interact. So we know there's some false positives. We don't really understand why that's happening. My team's trying to write a research paper about why this could be happening at the moment. The other thing is this is a protein. This is actually the protein that was shown in my first slides that was walking up the channel. It was carrying a big balloon on its back. It's called dynein. It carries a vesicle of chemicals around the cell. So it walks along tubulin kind of tubes. So tubulin assembles into the cytoskeletal proteins and it walks down those like tracks. This is the leg and this is the foot you were looking at before, and this is the bit that grabs hold of the big vesicle. So it's a very long, thin protein, but AlphaFold often curls it up. Now, as a structural biologist, I can look at that structure and go, oh yeah, those helices should be straight. So having structural biology skills help me interpret that correctly. But if you didn't know that, you might think that the protein wasn't stretched out, and that may impact your interpretation of the data. So it can still make mistakes. So now I want to tell you about the most impressive thing. So remember, a couple of years ago, you couldn't calculate a structure, and now we can calculate them. And I hope I've convinced you that it's actually doing a pretty good job and it's a useful tool. If we couldn't even solve the structure of proteins, how do you think we would go at making a protein? Completely de novo. Like, we've got the genetic tools to do it. I can program a bacterium to make any protein I want, but could I control the shape and function of the protein? It's a pretty big ask, right? If you can't even work out what they look like, could we ever engineer them? And this is where the new kind of generative AI is really, really important. So generative AI, you'll see, is used a lot in image production, so Midjourney, DALL-E, those sorts of things. So it's used to create new content. So this is the thing that people really get angry about on social media, about it's stealing stuff, it's making stuff. But in this particular case, it's doing some really amazing predictions and making stuff that I would say is something we should be really celebrating. AlphaFold 3 has a generative unit in it. That's what enables it to calculate structures of drugs and of nucleic acids. There's a limitation that it can hallucinate. So if anyone's heard about AI hallucinating, AlphaFold 3 can hallucinate, so you need to be a little bit mindful of that. It's all based on these diffusion models. So I told you about the first paper, attention is all you need. This is the second most important paper. This is denoising diffusion probabilistic models. So any uber machine learning pro geeks should read this paper. It's tough going, but this is the one that makes image generation and generative AI possible. So these are the two big papers I think of the century. So it starts with random noise. So the idea is that you're training a neural network, you take an image and you put a bit of noise over the top of it and you keep adding noise and it learns to go backwards from that step. So you say, okay, let's start with some noise and keep removing noise and then we get an image. So I'll try to show you some of how that sort of might work. So in protein space, we've trained it on protein structures and we keep giving it some noise. So we're going this way. So here's a protein structure and we add some noise and then add more noise and add more noise and add more noise and we keep adding noise until we got little dots. So we've trained the neural network to now start with dots and then it knows, oh, I've seen proteins, I'm supposed to make something that looks like a protein, I'm going to build a protein from noise. It sounds wacky, right? But this is how Midjourney and DALL-E work, same idea, and this is how protein design works. So you start with this denoising concept to build a protein. You get the backbone of the protein, and then we have another sort of neural network to add the side chains, and then we need to validate that experimentally. So that's the pathway to protein design, but it works. So there's 4 different types. You take some noise, and you tell it to make a protein. Okay, pretty basic, right? That's the original story of what I told you, right? So we can completely make a completely de novo protein that we can make in the lab, fold it, overexpress it, purify it, solve it experimentally, and it works. There's this thing called binder design. You take a protein that we know and love, let's say hemoglobin, our friend, and we tell it, well, let's put a little bit of noise next to it, and we can design something that will specifically bind to that part of the protein. So this is like what antibodies do. But we can program it to bind to a specific part of a protein of our choice. And then we can just make that in the lab. We can tell it to say, I want this little part of the protein, but I want you to build something completely different. So maybe this thing binds, but I want to make a totally different protein on one side. And the other option is we start with the protein and we just do a little bit of diffusion, and then we go backwards again and we can sort of rejiggle or remake that protein. So some examples. Completely new, never seen before protein structures, completely made by just going from noise to a protein, and they're totally made, totally stable, totally structured. What the? What the? This is nuts. This is binder design. So if we watch here, we're going to see the diffusion process. So this is the noise, and this is how it starts to build a binder. So then if we make those in the lab, that protein will bind to that helix. So we can choose now to make a protein bind to anything we want. And this is why David Baker was the third recipient of the Nobel Prize because his lab had been doing this. So why would they be useful? Mini proteins, like there's lots of different reasons. We can make lateral flow tests. We can make programmable engineering tools. So I want to give you an example of how a lateral flow test is. So what is a lateral flow test? It's a rat test. Who's done a rat test? Do you want to know how they work? They normally use 3 antibodies. Antibodies are difficult to make. We normally have to make them from mammalian tissue culture or from immunising animals. So there's animal research involved. We have to purify those antibodies. Then we then have to glue them to the test. So in this case, we can completely get rid of antibody production and we could make binders in the lab. So we would need to have 3 binders that recognise your protein of interest. So let's say tomorrow a new nasty virus just pops up, you know, at Sydney Uni, right? And we discover it's an ugly protein and we want to be able to identify it really quickly. So we can grab the person who's sick, we can swab them, we can do a DNA prep, we can sequence them. Overnight we can have all of the proteins from them and we can work out which is the coat protein of the virus. Okay, so then now I can use these binder methods to create 3 binders that specifically bind to the surface of that virus. So these are our binders. We glue a dye to one and we glue, we engineer on it a sticky spot that enables us to glue it to the experiment. And we also need one, and this is for the control, that very selectively binds to the first protein. So this is our viral protein, and these are the binders we're going to make. So this is our rat test. I hope it looks like a rat test that you remember. So we're going to take a sample of your snot. That's normally what we do, up the tube, up the nose, right? I'm sure we all love that. That was a great experience, wasn't it? So there's our little protein of our new virus, and we're going to drop it onto the test. And in the first well, we're going to put that first binder that has the dye. And in the second well, we're going to glue on a second binder. And in the third well, we're going to put the binder that binds the first one. So how is this going to work in time? We've got our proteins, we've got the dye molecules. This is how they bind to our protein. And this is what will happen to the first binder binding to the second binder. So let's work through what's going to happen. So we start with our protein of interest from your snot. It hits the deck and then starts to diffuse using kind of capillary action or diffusion. It just starts diffusing up the paper. It's just like cellulose membrane. Anyone done any TLC separating chromophores from plants in year 8 or something? Anyone separate dyes out and you see streaks? Yeah, TLC technology, same sort of stuff, right? It's just going to run along the deck. It's going to hit the first well, and the binder that has the dye on it is going to bind to our new protein. But it's not tethered, it's just going to keep diffusing up the deck. So if you remember your rat test, there was that pink dye, and you'd see it kind of move up the deck, right? So that's your snot protein, the COVID protein's bound to the dye, and it's now moving up the deck. In your rat test, it was an antibody. In this test, we can create these really quickly of a designed protein. It hits the second well. The second well has that other binder that binds to the other side of the protein and holds your protein that's now got the dye bound to it. That's the test well. So if a red line appears, it's telling you that there's a sandwich protein in between. So your protein's been sandwiched in the middle. So you've identified your protein, but then we still need to make sure that the test is working. So if there's no protein there or it doesn't show up, is there something wrong with the test? So we then have an excess of this always, so it's lots and lots. That's why there's lots of pink dye, and that's why you see lots and lots of pink initially, and it keeps flowing up the deck. So then those extra molecules that contain the dye then need to continue along to hit the second binder, and that's telling you that the dye binding system is working, the dye is present, and that's your control line. So I hope you all know how a rat test works now. So the advantage of binder technology is we could do this really quickly, we can do it cheaply, we can make it specific, we can do it without any animal research anymore. We can do multiplexing, which means we can mix a whole bunch of dye binders that recognise a whole bunch of different proteins from 10 different viruses to make a rat test that will identify 10 different viruses in the same test. That can make medical diagnostics really cheap. Cheaper, faster production, rock solid stability. So rat tests go off. Using binders, we can make it much cheaper, much more stable. You get really clean, the proteins are small, they're much smaller than antibodies, so they diffuse through the test very quickly. You'll get a very, very sharp line, not that fuzzy one. I remember how many people had like weak ones and you're like, is that a line? I don't know, it looks a bit, I don't know. You get much sharper results. They're easy to work with. We can design them to glue very specifically to the chip. And we can make it really fast. So if you've got a new version of the flu, a new virus variant, we can engineer these really, really quickly. So we only need to sequence a new COVID virus and we know the proteins change, we make a new binder overnight. So the test can change really fast. The ethics and IP of these are better. Most antibodies have been patented by biotech companies. So you can make a completely new binder, completely separate to anything that's been patented. So you can get around all of these expensive costs. And yeah, we can multiplex, so you can do this all at the same time. So this is what binders can do for us. That's why we care. All right, so now I want to tell you a little bit about protein enzymes. So enzymes are really just proteins that do chemistry, and your whole body's, like the whole function of biology is based around these enzymes. They basically have some amino acids at very specific places which enable them to do some chemistry. And if we could engineer them, then we can control biology. So this is super important stuff for tech. So there's this thing called the active site. So the whole protein molecule is scaffolded so that it puts a couple of amino acids at a really specific location. And that's called the active site of the enzyme. And if we could engineer proteins that put amino acids at exactly that location, we could make enzymes do anything we wanted. And that has just been total fantasy for my entire life. But we can now use this diffusion method to do it. And the Baker Group have done that. So here they've made 5 completely brand new enzymes using this sort of method. So in this particular case, they've been looking at motif design, so they lock in the residues. They use diffusion methods so that these are fixed and they do the diffusion around it, and they can make completely new enzymes that function with activity and are soluble and they can make in the lab. Like, really? This is crazy. Okay, so this is motif scaffolding. Why would you want to do it? So you could make enzymes that break down plastic. Okay, your laundry detergents already have some of these proteases. We can make the proteases better, faster, cheaper. Lots and lots of industrial processes, we can get biology to do it really quickly and cheaply. So you can make enzymes on demand to do the tasks you want them to do. We can make completely new tasks, things that chemistry has never been possible. We can make better medicines. We can make custom pathways. And I've got some colleagues at UNSW who are working to use these to clean up contaminated sites. So it's using biology and chemistry to control what's happening. Obviously, and we can do all sorts of cool, funky things in the lab to do fundamental science things. Probably not so much of an interest to you guys. Okay, and the last one, I think this is the last one, is partial diffusion. So this is kind of just putting a little bit of noise in to kind of wiggle it around and then come back with a new structure. And by doing that, we've been able to build completely new medical cases where we can make very specific, tighter binders for therapeutic treatment. So this is a big problem for us. So we can use this methodology to make things bind more specifically and tighter. So that's a better drug. If it doesn't bind specifically, you get off-target effects. It hits another enzyme, and that can make you really sick. Okay, and this one's really cool, is they mix all of the methods together, and this one just came out in March this year. They basically use this technology to make specific binders to some snake toxins. They made all the binders, and they demonstrated that they can completely cure envenomation from a snake using this method. So there are plenty of real-world examples where we've only had the technology for 2 years and we've already been able to make it work. So this is the reason why, this is all Baker Lab. This is the reason why David Baker got the Nobel Prize with the same kind of team from the AlphaFold. So this technology still uses folding in the background to do the diffusion as well, but together these methods are really powerful. And this is going to be the future of biology in wet labs. is you can specifically make a protein bind to anything you want. It takes months to raise an antibody, and that's the only way we could specifically target a protein previously. So what I wanted to tell you is that the technology is advancing quickly. It's a really exciting time to do molecular research. Great care is needed. It isn't magic. It still makes mistakes, so you need to understand what you're doing. But not all AI is bad. This is the single most exciting thing I've seen in science in my life. And then back to my team again. So that's the end of it. We're done.
I said it almost dismissively. Like, thank goodness for that. Like, that's awesome stuff. We have time for some questions. Thank goodness.